All right, we're officially starting our class, our last week of classes happening right now. And so here we are. I have your third exam. I'll hand that back at the end of class. So we'll talk about that some today, and we'll move forward. And so this is where we're at, what you should do this week. So I will put the answer key video on the Brightspace, and it's important to go through that and watch that. And then go over your exam, and remember that the way I grade exams is I rarely put down comments. Sometimes I write down okay, and when I write down okay, I just mean that's an okay answer. It's not the best answer, but it's an okay answer. And I just want to put that down sometimes. But mostly, as you see, I'm just marking points off because I'm also taking notes off on the side about what I should talk about in the answer key video for everybody to see. And no matter how well you score on the exam, high or low or in between, you should go through the answer key video. I'm making it as a resource. And that's because we're still learning about the style of questions, the expectation for the answers in a general sense for our course. Because our final exam is going to look exactly the same as the first three exams. The questions are going to be the same style. It's not like I'm going to suddenly say, well, just remember like anything you can tell me from the course, or a general, answer like that. Nope, it's just a bunch of specific stuff. And I emphasize, when we were preparing for our third exam, we started preparing for the final because, I have more questions that could have been on the third exam and could have been on the second exam and could have been on the first exam. And it's important to go over all those exams to set yourself up, to remind you all the stuff we've learned, because it comes back for the final. And so that's what we're doing. And so we can spend time this week talking about questions you have. I would love to talk about new material because this is my favorite topics of all time in my life, like that. And there are no more assignments, no more reading, but today and Thursday we'll have new material given the opportunity. So that's where we're at. And the final is, I guess, a week from today in here. Okay, like that. Okay, so that's where we're at. So I have the grade distribution for the answer key video for the third exam, and I'll post that after class with the recording from today, so you can see what your letter grade is. And I actually haven't had time to make the answer key video yet, but I plan to do that in between classes today and get that posted. But I know you're especially interested in seeing the grade that you got. And so that's the same story as in previous exams, where you're going to be represented as an anonymous asterisk, so you can see your score corresponds to that grade. And overall, people did a good job on this exam. So on the syllabus, the value for the exams goes up a little bit, because I've seen students' performance goes up a little bit, and that's one way to help support that goal, because that's a big goal of college, is improve your exams in each course as you go forward. So the final exam is, no matter where you're at in the course, the final exam is, is it worth like 35, like a third of the grade. And there's a movement in assessment grading nationwide called grading for equity. And equity means fairness to everybody, coming from different backgrounds. And the idea is simply, should not have a big emphasis on the first exam because people are coming from different places. Should have emphasis at the end of the course because everybody got a chance, good chance to catch up and figure out where we're going, what we're doing. So I know that's rough because you have other final exams and assignments and stuff too, but yeah, I think it's 35% of our final grade is based on that final exam. Again, it's going to be the same format, questions like that. Okay, so that's where we're at. So I just point this out. There was this 3C technique question that Kaylin asked just before the exam, and I'm like, what the heck is that? So I'll talk about that I said I'll follow up on that, and it's a good question. It's good technique to talk about for us. So we can get there and do a little of that and some more stuff, but let's just see if anybody have any questions right now about anything at all. Yeah, Dornica. Sorry. One time I was closing a window like this and I yelled out of it, and there were people down the hallway that were, this is tough, but they were like triggered. You're in a school classroom building and somebody's shouting and they're like, ah! Like that. One of the instructors came running down. I'm like, I'm so sorry. just yelling out the window. So anyway, Dornica, what?
Is the exam also 50 questions?
Yes, same style. They get kind of bundled together a little bit like this and that. Yeah. Yep. And other things, anybody wants to talk about Don Diego?
Starting at 8 and ending at 10:45?
It's officially 8 to 10. I don't have anything after. And so I said before, 8's pretty early for some people with their schedules. And if it happens that, you're trying to get here at 8 and you don't get here at 8, don't just drive crazy, right? Just get here, come in, the setup, quietly, sit down, go to the bathroom, turn off your cell phone, put your cell phone out, keep everything off the table, sit, far apart from everybody and just quietly get it done. And, it's the same style. We've done the other exams in 70 minutes, 70, 80 minutes like that. So, 2 hours is plenty of time to do it. But, if somebody comes in at 9, they can take it till 11, like that. And, it's good to try to get here and get going. But again, I know, whole semester you've been trying to get here at 9:30, and that was hard, right? It almost went to 8 a.m. and you're like, oh. Yeah, but yeah, so just come on in. This is where it's happening. And yeah, you'll have a fair amount of time to do it. Yep. Yeah. Other questions? Yeah, Bryce?
Is there any emphasis on the more recent material in the final, or is it?
Well, luckily so far, yeah. And like I've described to you before, I knew what was going on in college. I would just clog up opportunities like this with questions about like, well, what's your favorite pet you've ever had in your life, instructor? Like that. Oh, yeah. Yeah, right? There's no new material yet, right? Yeah. And, but no, kind of a natural balance, it's just the best stuff like that, you can ask questions, but we really want to get more of this. Oh man, I love these topics so much. Yeah, so yeah, not a special emphasis on stuff we haven't even gotten to yet, but some definitely, yeah. And it fits in, right? It all fits together, so yeah. Yeah, other questions people have, anything at all? Yeah, Sophie?
So you said to be able to make connections throughout the units that we've gone over?
No, it's not really that. It's not going to be, you're not going to be asked, sorry to interrupt, but you're not going to be asked to kind of synthesize, put it all together like that, But you are going to be expected to use examples from, the other part of the course that just fit in with explaining, well, what about this, like that. Like I'll tell you that 3C technique, that's chip. We learned about chip for our first exam. It's just chip using a different purpose. So that would be a good example if we get to talk about chip, if we get to talk about 3C. There's a reason, people are doing 3C, but it's chip. So we better talk about both of them. And in talking about them, we should talk about the overall strategy, because it goes back to your question earlier in the semester about binding and what's binding, what's that like? Yep. And yeah. So yeah, not a thing where you're going to be expected to pull it all together and stand up on all of it and be like, I see something new I can explain, but just connect those examples. Yep. Yeah.
Can we also review the previous exam that we had for the final?
You mean in class?
No, at home.
Oh, you should. Yeah, definitely you should. Yeah. Again, I put a lot of emphasis on the idea of, the mindset of expectations, of how to see, because I've had some students in classes in the past, and they've had me for multiple classes, and they have turned around to the class and say, is what he's doing. All the time in class, I'm telegraphing questions and answers. You know, I'm not really intending it, but I was a student and I want to know what's going to be on the exam. And so limited time in class, just talk about stuff on the exam, you know, like that. So I think it's good to go over exams just to get in that groove of, okay, it's going to look like this, you know, and I'm going to expect it to spit that out and not just go like true or false or, you know, multiple choice, but actually make a statement about that. Yep. Yeah. And another thing is sometimes, you know, when you go over old exams, the instructor will put a couple questions from the old exam on that, you know, just to encourage it. That could happen. You know, I don't want to because I have a lot of other questions, you know, to ask. But then another thing that can happen is ask it in a different way, you know. And again, I really think the purpose of those earlier exams is to get used to that, and then go back over it, know that material. You know the material from those exams. That's, you know, a big goal. Yeah. And so you can, so today and Thursday and anytime in any of my classes, you can interrupt anytime. You can just be, I have a question. And it can be way out of context. You know, like my favorite pet, Chopper. Our dog when I was 13, you know, kid, we got a dog named Chopper. Man, what a pet. Like that, right? You know, and no, Bryce, that's not going to be on the file. But dang, Chopper, oh gosh. I miss Chopper every day almost. Yep. All right, so let's talk a little bit about this 3C technique. And all you have to do is say, oh, that's chip. So let's talk about chip. And in talking about chip, we will talk about whether the purpose of chip is and how you do it. And then add that 3C technique. And then there's one other method we'll add called clip-seek. And they're all the same thing, just different purposes. And all of them are based on the original chip thing we talked about, which is, remember we learned like 5 methods where you're studying a gene and you want to identify the enhancer. So you can do a deletion study, you start cutting off pieces of the DNA near the transcription unit to map where the enhancer is located. Because if you delete the important regions of the cisregulatory region, the gene won't get expressed. So identify where the enhancer is, then do a nuclease footprinting. Is there a protein binding to that enhancer region? If so, it would protect the DNA from getting degraded like that. Then do a gel mobility shift assay. If there's a protein that will bind to it, it will slow down the mobility of the DNA in the gel. And then build an affinity column, so beads with the enhancer DNA attached to it, and flow the nuclear extract over it. Your protein will bind, all the other ones will flow through, and at the end you can release your protein, and you've purified the transcription factor that is the binding to that enhancer. And that's how scientists would have found lots of these transcription factors. So we talked about CREB, the cyclic AMP response element binding protein, right? They started out going, you know, well, we see cyclic AMP going up in these cells and there's cell response. I wonder if it includes change in gene expression. So let's hunt for genes that get turned on, and they're like, they do. And they're like, what's that transcription factor that activates this gene? Because it's sensitive to cyclic AMP. And so they chased it down using those kind of methods, and there are more, but those kind of methods, and they got that, and they said, we'll call it CREB, cyclic AMP response element binding protein, because that's what it is, right? It responds to cyclic AMP, and it binds to a response element and enhancer in target genes that get expressed when cyclic AMP levels go up, like that, right? Or that serum response element binding protein, you know, most of these transcription factors, many of these transcription factors were identified by that purification strategy, okay? So then we say, okay, we purified that transcription factor, right? We had the beads and the enhancer DNA bound to them, and we sent the nuclear extract over it our transcription vector bound, we released it, we have our purified transcription vector. And then a really common method that opens the door for these activities is we take that purified transcription vector and we inject a little bit into an animal, like a mouse or a rabbit. That animal's going to respond to it, and it's going to make antibodies. Because it thinks it's like that protein, that transcription vector, the animal thinks it might be a virus or part of a bacteria. So it's going to make an immune response to it, and we've learned in our course a little bit about the immune system, and antibodies are proteins that are made by B cells, and they just stick to targets. And so that's the idea of, we're going to make an antibody, and the antibody will stick to our transcription factor, and the example we used was the estrogen receptor. So now we're going to study it, and that's when CHIP comes up, because we can use that antibody to grab hold of the estrogen receptor and pull down all of the enhancers, all of the estrogen response elements, all the target genes. So CHIP is a method, right? What is CHIP used for? Identifying all the target genes of a transcription factor. And from that, we discovered that sometimes the enhancer sequence, the response element, the sequences vary a little bit. So it's hard to find those parts of the cis-regulatory region because they don't all look the same. But you can do a functional assay like CHIP and pull them all down and identify all the target genes. Yeah? So then the next thing to say about the CHIP method is, well, to use the antibody, you're going to have to break the cells open. So you're going to have to take some cells, probably initially isolate nuclei. So remember, we can use a Downs homogenizer to break the cells, but not the nuclei. Pellet the nuclei. We got the nuclei. Our antibody can't get into the nucleus, so we got to break that open. And that's going to happen in a test tube, and there's going to be a little detergent around, and scientists worry that there could be some false binding. And they call that an artifact. It's just, part of the method is we're going to break the nuclei open, and we're going to have this nuclear extract, and the estrogen receptor might suddenly bind to other DNA. That isn't a true response element binding, right? And so, because they're concerned about that, they invented the first step in the method. And the first step in the method of CHIP is to use just the living cells are just happily there, and you add formaldehyde. And formaldehyde is a famous preservative. It's a little tiny molecule that's chemically reactive that will form covalent bonds between biological macromolecules. So it can cross-link protein to DNA or protein to protein or protein to RNA, like that. And it does it really fast. And so the living cell doesn't see it coming, and it's a preservative because it will kill the cell. It freezes the molecules in place. And you do it for just a short time, so there are just a relatively small number of these covalent crosslinks. That allows you to do the nuclear extract with lots of detergent, because you're not worried that the proteins are going to get denatured. You're not worried that the non-covalent binding is going to get disrupted. You want it to get disrupted. And you only want to preserve the situations where in the cell, the estrogen receptor was really right next to the DNA, because formaldehyde is a little molecule. And it was right next to the DNA, and it got covalently cross-linked. And so that's a key concept, that lets scientists be confident that in the living cell, the estrogen receptor was bound to that DNA, because it got cross-linked. And the formaldehyde is a little molecule, so it can only cross-link things that are really touching, like that. So it's a great strategy, because it really reduces the concern about, after you break the cells open, having false binding occur, because you do it in the presence of detergent and other molecules that help disrupt non-covalent binding. So then I'll just draw this out, because it's also kind of cute, is we learned about this, making a tube like this with a mesh bottom and then having beads in it, and we have this thing where we could put enhancer DNA on covering those beads, and we could use that as a purification strategy. So that's going to be enhancer DNA, and we're going to purify this to back up in time to when we're just purifying the estrogen receptor for the first time. And that's a cool strategy. The strategy that's really common in CHIP is that instead of having the antibody on beads and grabbing the estrogen receptor and the cross-linked DNA as it passes by, what people do that is just very cute is they'll put the antibody on beads just in a test tube. So that's the antibody, and we're talking about estrogen receptor, so the estrogen receptor antibody, and let's say we made it in a rabbit. So it's just a rabbit antibody from the blood of a rabbit we collected and purified, and we have it on a bead, and it's just in a test tube. Okay, and so then we add our nuclear extract, and we have some estrogen receptor bound to some enhancer, like that, okay? And that's what we're interested in that enhancer DNA. So instead of running it over a column, they just put it in a test tube and shake it up. And then the super cute thing is this bead has iron in it. And they use a magnet to pull all the beads over here, and they wash away all the other stuff that's not bound. And it's a cute method because it avoids some of the issues of centrifugation and or running passing cells over this column like that. And just a detail, but the idea is you've got that cross-linked complex, and you're going to use the antibody to separate that. And then after you've done that, it's possible with heat to reverse formaldehyde crosslinks, and you can release that DNA, and you can analyze what the DNA is. So that's CHIP. What is CHIP? The purpose of CHIP is to identify all the target genes. And remember, like that E2F transcription factor, it has hundreds of target genes, like that. You want to know all of them, because they're all involved in start, like that. MYC has a bunch of target genes. Then there's some transcription factors that only have a few, but you want to know what all of them are. Because what does myo-deactivate in addition to myogenin? If you're studying muscles, you want to know, like that, because they must be really important for muscle stuff, right? So CHIP is super awesome, like that, okay? And so 3C is the exact same thing, but you're just trying to find out something different. So here, CHIP is, remember, the purpose is to identify the target genes. In 3C, it's exactly the same method. It's CHIP, but the purpose is to identify molecules that are close to each other in the nucleus. So 3C came up when we talked about what's going on inside the nucleus. Can we look, and Chase brought up, they talked about this fish technique at the same time. Where are the different things located in the nucleus? We learned about some bodies and stuff like that, okay? And so here's the deal with CHIP, with 3C. And it's the same idea, but this is what people want to study with 3C. And it's how is the nucleus organized, but some of it is down at a close level, because they're using formaldehyde as a cross-link, so these have to be things that are touching, right? And so what's a good example of that? So what's a good example of 3C? Oh man, well, how we talked about how the enhancer can be far away. It has to be cis, but it can be far away because it's a tether, it's a leash that's holding it. And so then we said, you know, this transcription factor, like heat shock transcription factor, it's gotta get to the arrested polymerase to release it. Right? So that's the rate limiting step in transcription. We gotta dig back, right, and remind ourselves that we learned about this junk, you know, back in October. And so there's gotta be some touching going on here. So who's touching what? Right, like that, right? And like MyoD's touching Tada, right, like that. So you can use this 3C to identify who's close to each other in the nucleus. And you'd have to have an antibody to one of the molecules you're interested in and say, who's touching it, you know, who's it touching, like that. So it's a really good technique. It's weird that the authors put it, it's partly weird that the authors put it right there, because the other things we were talking about were bigger neighborhood stories, not who's touching who. And the reason the authors put it there is if we look at DNA, well, there's also these histones. So, you know, what about those histones, right? So that's a 3C thing that people are interested in. Also, you know, how are these histones organized into the nucleosomes? Because the nucleosomes fold things up into little circles like that, but then they get organized into sort of spirals of nuclei to further pack the DNA into the nucleus to make it shorter. And then those spirals sometimes have loops like that. So that's called a chromosomal loop. And just to show you, that chromosomal loop might have 2 or 3 genes on it. So we're talking about a longer piece of DNA. And, you know, and this is where the 3C was talking about who's touching who at, you know, the start of all these genes and regulating transcription. But the 3C people also study these bigger loops to try to understand that organization. So who's touching who here at the base of the loop, you know, to make a loop. So it's, you know, kind of partly weird that they put the 3C technique there, because it's a really zoomed in technique like that, whereas they were talking about just neighborhoods in the nucleus. And this is why they put it there, is because in addition to the, I don't want to call it a loop, but the tether bending for transcription activation, which is something, you know, we're right on that, right? There's also this a little bit zoomed out a little bit to show some of the chromosome organization like that. So that's at 3C, it's chip like that. Yep, okay, great. And you gotta have an antibody, right? So 3C, what do you need? You need an antibody, and you're looking for things that are touching each other in the nucleus. And you wanna make that cross-link before you break the cells apart. Same exact method, just a different purpose. So it's great because we can mention ClipSeq also. So ClipSeq is chip also, but they don't want to call it chip because instead of looking at chromatin, chromatin is the DNA and the proteins that are bound to it, ClipSeq is the same chip method, but you're looking for what proteins are bound to the RNA. So you're studying SNRPs and splicing. You're studying those proteins that are always bound to the RNA, that proteins that need to be bound to the mature RNA for it to leave the nucleus. So that's just that. ClipSeq is people studying proteins that are interacting with RNA. And it's got a different name, but it's the same method. So, you know, I would say, oh yeah, it's chip, but it's because it's the method that's the same. You gotta have an antibody and then do that same cross-linking like that, okay? Questions about that? So good stuff, you know, to follow us up on, and a little bit of new stuff, right? We could describe 3C, chip, and ClipSeq on the exam. three methods where you have to have an antibody and you're looking for direct contact using cross-linking strategy. I'll just show you this. Oh, I didn't get that slide in. Does it show this? Yeah, just a side note is when people do ClipSeq, that's the newer method. And with ClipSeq, it's just cute to hear this. And you don't need to know this, but we talked about formaldehyde. With ClipSeq, they use UV light as the cross-linking agent. So you know that UV light can cause DNA damage, and it causes it by breaking covalent bonds in the nucleotides, and then new bonds are formed, and it's damaged the DNA. UV light does that to lots of different types of covalent bonds in macromolecules. And so you can use it as a cross-linking agent also. And it's really amazing because the cell does not see it coming, right? With formaldehyde, you do have to add it, and the formaldehyde diffuses into the cells, and the cells are like, wait, what? Really fast and very short crossing agent. UV light is a zero-length crossing agent. They gotta be bound together. So you can use UV light as a crossing agent for CHIP or 3C or ClipSeq. It's just a dramatic example because the UV light sources they use are like almost like lightning. They're like, you know, like a photo flash of just really high energy, just like kaboom. Yeah. So just cute addition to that, Anna. Okay, great. Okay, so that's those methods like that. And questions about those? So we have a nice package of those because that came up, right? Well, we want to know about the, you know, the touching, right? How did people find out what MyoD touches to get through the rate limiting step? What does HSF touch? You know, it could touch some part of that resid polymerase like that, you know? And so good method to add to our stuff, you know. Proteins are always bound to RNA. That's, you know, that's always the case. You rarely hear that. And then they're important in splicing and polyadenylation, stuff like that. So, you know, good methods like that. So there are some other methods that are used to identify those direct interactions. So that direct binding, that non-covalent binding, like that. And one that we should talk about in our course is called a yeast to hybrid screen. And it's from 30 years ago, but it's just, it's very, very effective, and it's just cool to hear about it like that. And so we have to start out picking some binding, and it's for protein binding only. So the others, ClipSeq was protein RNA. The CHIP could be protein or protein DNA. Yeast 2 hybrid is specific protein interaction. And you know, we had the question about binding. You know, how do we know they're binding like that? And so we'll use for our yeast 2 hybrid screen, we'll use just as an for example, insulin, that's a protein, and the insulin receptor. No, we shouldn't use that just because people ask questions about it processing. Let's use PDGF, it's a protein, and the PDGF receptor. So we say, you know, we've got this thing that gets released from the platelets, PDGF, and we've learned that it increases the mitotic index of skin cells, right? So we're dialing way back in time where we're like, I wonder what the receptor is for this PDGF protein. There must be a receptor on or inside these skin cells for them to respond. So let's go fishing for the receptor, like that. And that's what the purpose of yeast hybrid is to identify protein-protein interactions exclusively, like that. Very different method, protein-protein interactions. And it goes like this, and you'll see it's great because it also loops us back toward the start of our course and being like, remember, we learned about this, right? So it goes like this, where this is just the yeast that causes bread to rise, that yeast species, Saccharomyces cerevisiae. And back in the day, Nobel Prizes in medicine were awarded for scientists studying this. It was very hot organism to study. So scientists at MIT and Harvard and so forth were studying this yeast because it's a single-celled eukaryote. So it's got some, relatively close relationship with us. For example, all those cell cycle markers that we have to, or regulators that we have to remember for the final exam, almost all of them were studied in yeast using a genetic strategy. Sidelight to that is that some of those labs, like there's a lab run by an old guy at MIT who still study yeast, and they've had resurgence because of other topics that they're relevant for. And his lab very famously studies aging. And there are people who take dietary supplements based on the role of those in yeast aging. Yeah. His name is Leonard Guarente. There's a professor that was his former student, David Sinclair, at Harvard, and there's lots of YouTube stuff about David Sinclair explaining how to slow down aging by following this research that was done in the yeast that causes freaking bread to rise, like that. Yep, so yeast. So yeast, was like a heyday, and then kind of faded, because they started looking at, fruit flies more and stuff like that. So this is this yeast 2 hybrid. And so lots of genes are understood really well. And there's a transcription factor in yeast that's called GAL4. So this is going to be a GAL4 story. And it's a transcription factor, and it's got a DNA binding domain and a transacting domain. So DNA binding domain and a transacting domain. And so remember, right, it's going to bind to an enhancer. So here's a target gene, and there's an enhancer, and it's a GAL4 enhancer. Like that. And it's very well characterized, that transcription vector. In a really simple single-celled organism, you can grow a lot of yeast overnight in a big vat, like that. Okay? And so the strategy goes like this. Well, there are these 2 domains. They have to be together, right? You have the transacting domain. That's going to affect the rate-limiting step in the target gene and increase transcription. So this has got to be there. And the DNA binding domain is what is tethering it, right? We got to tether it. We're going to tether it to this cis regulatory element, and the DNA binding domain hangs on. And what it's hanging on for is to have the transacting domain stay in the neighborhood, like that, right? It's like you're holding the biting dog by its tail, it's, I don't know, that's not a good one. But they're physically connected, like that, okay? And if you had a mutation where either one of those is missing, it wouldn't work at all. They have to be physically part of the same protein, okay? And then we know there has to be a GAL4 gene over here, right? Because all transcription factors are proteins, and there has to be a GAL4 gene, like that. And in the coding region for the GAL4 gene, there has to be, part of the coding region has to be for the DNA binding domain, and part of it has to be for the transacting domain. Okay, so that's just what's naturally occurring in yeast. We got a transcription factor. It's just a well-characterized transcription factor. It binds to cis regulatory elements, and there are target genes. Okay, so the scientists start with that, just natural yeast biology that they understood really well, and then they start building 3 transgenes and put those in to do the yeast 2 hybrid assay. Okay. So here are the 3 transgenes. They're going to make them, they're going to put them in yeast, they're going to just be extra genes in yeast. The yeast is going to be totally normal, wild type. It's going to have normal GAL4 doing its thing, and it's going to have these 3 extra genes. So one of them is, well, let's probably erase this and draw them out. Okay? So one of the transgenes is, these are the 3 transgenes that we're going to put into yeast to do our assay. So one of them is a GAL4 target gene. So it's going to have that GAL4 binding site, and we are going to replace its transcription unit with GFP. So if this gene gets activated when we put it in the yeast, the GFP protein will get made, and the cell will glow. So, you know, we're familiar with that, right? We just swap the transcription unit with that regulatory region, and we put that jellyfish gene transcription unit into yeast under the control of GAL4. Okay? Oh, you know, and I quickly said the yeast is wild type, but the yeast has to have a mutation where it doesn't have its normal GAL4, because the GAL4 would just activate it. So we don't have the normal GAL4. What we have to activate it with is the 2 transgenes. So the other 2 transgenes that go in look like this. So both of them are GAL4, but one is GAL4 that only makes the DNA binding domain, and then the other is GAL4 that only makes the transacting domain. And if we put those 3 transgenes in, the cell would not glow, right? Because we will get made the DNA binding part, and that would stick here, but it doesn't have a transacting domain, because the transacting domain is made on a different gene. It's a different protein, and it's just floating around. So that's the system. The experimental system gets set up like that, where we don't get glowing cells because we can't activate the target gene, because the only part that's binding is the DNA binding domain. Okay? So then the idea is this. All right, so we start our experiment with this, and we're studying PDGF. So we've seen this before too. We take this, and to do our experiment, we add the PDGF coding region to this gene. And now we make a trans protein looks like that, where it's got PDGF stuck on the target gene. And that's not going to glow. PDGF has nothing to do with transcription, right? And this gene and protein are called the bait, because we're going to go fishing for something that would bind to PDGF. And we do that by inserting randomly, I should do a little different color here. Here, we insert random coding regions. And there would be thousands of them, tens of thousands we want to insert. So we're growing this in yeast, and each yeast cell can have a different random insertion. And as a result, this transgene gets made, and in each cell, it has a different portion here, like that. And if we randomly happen to have the PDGF receptor in a yeast cell, those two would bind, right? PDGF and the PDGF receptor bind. And we would recreate the GAL4 transcription unit, transcription factor, because we got the DNA binding domain, and we brought the transacting domain to the target gene. So this is a genetic and molecular biology strategy for looking at protein-protein binding, right? And what if, we randomly inserted hemoglobin here? Well, that wouldn't glow, because hemoglobin does not bind PDGF. What about the insulin receptor? No glow, right? The only thing that's going to glow is having randomly inserted a coding region there that would bind to PDGF in the cell like that, in a natural way. And so they use yeast, partly because it's so well characterized, and partly because they can grow millions and millions of yeast cells and just have random genes in this transgene. And the bait is just trying to get it to bite. That's why they call it the bait, right? You're going fishing and you're hoping to get a bite. So that's a nice one to describe, just because we're so familiar with the parts, right? And a graduate student, a student like all of you, thought it up. They thought up, the transcription factor has to have the DNA binding domain and the transacting domain together. And if we separate them, it does not function. But we could design a system where they could get brought back together and the cell would glow like that. So it's good stuff. And it's nice because there are a couple of different strategies that do experiments similar to that CHIP, CLIP-seq, 3C strategy, trying to find who binds with who. And this is just a very different one, but it's all built on logic, back from our first exam, right? This goes back to, okay, I remember transcription, rate limiting step and stuff like that, yep. So just good, to bring that up as a new thing. And scientists found a lot of proteins that bound to other proteins that way. Like maybe FOXO. What binds to FOXO? Like that. And some of those are tricky because FOXO only binds to 1433 when it has been phosphorylated. And yeast might not do that, So there might be a post-translational modification that yeast doesn't do, and the experiment wouldn't succeed like that. So it's not a perfect strategy, but it's a pretty good one. And it's very sensitive because you can use like a light detector to detect just tiny amounts of elimination. So even if you get a little activation of the target gene, you can say, oh, looks like something's binding. Yep. Yeah, okay, so that's that. I just wanted to put that in because I think it fits in, and it helps us drive back toward the earlier parts of our course. And remember, those are really important. Most of our course really we spent on regulation of transcription. And that was, the third exam included more, so post-translational, post-transcriptional processing and stuff like that. Yep, questions about this method? Okay, so that's that, and just a good one to add. Doesn't really add, much, just that logic is applied to invent a strategy to find protein-protein binding. So now we've talked about 4 methods: CHIP, 3C, and CLIP-seq. Gotta have an antibody. It's all about having an antibody so you can grab a hold of the molecule you're studying. And then there also is cross-linking in the living cell. Then they just study different things. So CHIP is what are all the target genes of this transcription factor? That's CHIP. 3C is what are some contacts made when the enhancer binding protein activates transcription, or loops in chromosome. What are those touching? And then CLIP-seq, what proteins are bound to RNA under different steps, like that. Yep. Yeah. And then the different technique is the yeast-2 hybrid, which is a genetic strategy in yeast to go search for protein-proteins interactions. You've got a protein and you want to know who's binding to it. So a great example is PDGF and the receptor. Yep. so that's that. Okay, so then, so what we're going to talk about in any time we have left is in part 2 of our textbook, there are 2 chapters that we're skipping. You don't need to look at them, so we've talked about that in other points in our course. And chapter 4 is about genes and genomes, and chapter 9 is about genomics, proteomics, and system biology. And so what we're going to do is just add some to the end of our course about the scale of modern biology, because it's jumped up in scale, and it's good to bring our course to a close with that. And we can do that especially with genes and genomes. So we'll start talking about genes and set them in a little bit of a genomic context. Not really adding too much new stuff, but kind of going back and remind you of a few things. And so that's where we're at on that. Now, part 2 was called The Flow of Genetic Information, and we've talked about that. We did a great job pulling those stories together, right? Gene expression and the pathway of gene expression. And talked about transcription regulation. That was we read chapter 1, then we went to chapter 7. So, we have a good handle on that section. And just to look at the chapters in our book, I think I have them. Yeah. We read chapter 1. Chapter 3 was fundamentals of molecular biology, so we've been cranking on that along. Chapter 2 is chemistry. You get prerequisites for chemistry. You got a good chemistry program at USM. Chapter 4, big one for us, cancer biology. Part 4. And then part 3 was that cell biology that was really a big part of the 3rd exam. So we're doing a great job, covering most of our text, even though we didn't read every chapter, because we put it together, as efficiently as we could. All right, so we're going to talk about genomes and genomics, and that's the original term of the big data biology movement. And the idea was, back 25 years ago, 30 years ago, each scientist was studying their own thing. I'm studying MyoD. I'm studying, whatever, PDGF, and everybody's on their own, and they're trying to, get the DNA sequence and read the DNA of all their genes, study, focus, and folks realized maybe we should just team up and just try to get the entire genome, all the genetic information read and sequenced. And so the human genome was sequenced, and we'll talk some about that. And then that genomics and that omics term got applied to other big data strategies. And one we learned about already is this cute name, interactome. So 1433 interacts with lots of proteins. Let's find all of them. You're studying FOXO, I'm studying CDC25. Let's just get them all, figure them all out. I'm sure we'll learn more about our individual protein by finding all of them, like that. And so there are other examples of that. Transcriptomics is, let's look at all the genes being expressed. You've been looking at what genes, you're studying each transcription factor turns on those genes. I'm looking at estrogen receptor, what genes get turned by that. Let's look at all of them. Let's just look at all the RNA, we'll find out all the genes. Because for a gene to be expressed, it has to have the RNA made. So let's study the RNA. And it won't be all the genes. We won't detect insulin RNA in any cells except for the beta cells of the pancreas. The gene will be there, but we're interested in insulin expression. So that's that. Proteomics, we talked a little bit about that, and that is post-translation modifications, right? And regulation, is a big part of regulating gene expression. And it's hard to study because you got to learn all kinds of different methods, totally different methods. So we'll dive into some of those, and we'll start with this genome and genomics, like that, okay? And so in that, I just want to mention a couple of things. So the first is just a term, systems biology, then just an example of biotech companies, and then we'll talk about genomes and some methods to understand where we're at and what's happening in biology today. So the first thing is just this term systems biology, and it's what I was just talking about. Instead of having it be hypothesis-driven science. So for example, I hypothesize that PDGF binds to a receptor, and I'm going to test my hypothesis. So that's hypothesis-driven traditional science. One hypothesis, one experiment. Systems biology is, let's just get all the data. We'll worry about hypotheses later. Let's just get all the data. We've got methods that can collect massive amounts of data. So let's get the whole genome. And then you can start having your hypothesis, and Chase can have his hypothesis, and on down the line. We'll just have a bunch of data. And along with collecting the big data comes some big data thinking about, well, what are some bigger questions we can ask like this? Yep. So that's just a term, systems biology. And there's lots of systems biology in modern biology, and it really just means big data and then thinking about big data. How can we think about big data new, like that? Yep, and there's a lot of it, and it is a different, when you step up, it's a different set of topics to talk about. Yep, okay, all right, so systems biology, just that term. Then just to give you one example of biotech companies. So in the last 30 years, biotech companies have really increased in size and activity. So it's been a really long time since there was this idea of biotechnology. What can we take advantage of that we're discovering in biology to make products and stuff like that? So there are a lot of biotech companies that work that way. And to just give you one example. So this is the website of one of the two companies that developed the mRNA vaccines for COVID. And it's very interesting to talk about mRNA vaccines and COVID for a whole bunch of reasons. Here's one. Like when COVID showed up in the United States, when it first showed up, like 2 people had it. A month later, this company was testing the COVID mRNA vaccine in humans. And 2 months after that, Trump gave these companies $9 billion to do whatever it was called that he said, it's the magic bullet thing, or he had a term for it. It's does anybody remember the term? It was like a Star Trek term.
Oh, wasn't it like warp speed?
Warp speed, yeah, Star Trek term. It was 2 months after they started it, And they're like, that's cool, we're well on our way, we're pretty solid on this. And one of the companies is Moderna. It was a little biotech company in Boston, and the other is BioNTech, or BioNTech, and that's in Germany. And you've heard about the Moderna vaccine, and the other one they call Pfizer, because BioNTech was a small company that made it, and then Pfizer could manufacture and distribute it like that. So these are these two, and people talk about big pharma, and they're like, is that mRNA vaccine big pharma? And you're like, well, no, it's these tiny biotech companies that were working on this strategy. And I'm not, promoting, I'm not selling stock, but it's just cool to see it happen, because you can't imagine, if your life path had led you to be working at Moderna in Boston, because any one of you could have been just working there, and then they get like a few $1 billion from Donald Trump, like that, and they're like, all right, we're gonna get the COVID done. And this is just their website, and it says, I just grabbed these, it says the mRNA pipeline, right? So, mRNA vaccines, these are all mRNA vaccines, and it shows them, phase 1, phase 2, phase 3, commercial, like that. And so this is just a list, and they're hiding this, it's private, competitive stuff, but they're also just on their public website going, look out. So here's COVID, flu, like that, and just cranking them up. And then other viruses, Epstein-Barr virus, norovirus, like that, Lyme disease, vaccines against these, right? They're moving forward on a Lyme disease vaccine, other Some vaccines to viruses that aren't even, common in the United States, but worldwide vaccines like that. Melanoma, so cancer, you've seen there's been news recently about cancer vaccines. Let's get a vaccine to the cancer. And that's typically after a person has had the cancer and they've done surgery and radiotherapy and they think, we think you're cured, we're going to give you this vaccine to help you for any remission. And bladder cancer, metastatic melanoma, early and advanced solid tumors like that. More tumors, because of course there's big money in that. And so it's just this long list, and it's the same technology, and the technology looks like this. So they're an example of another term we can learn, and that's synthetic biology. And so that's where you're going to take advantage of what you know about biology and start building stuff. And so if you just look at this, you would say, what we learned is that looks a lot like the LDL particle. Very simple little particle, and it's an mRNA vaccine. And mRNA vaccines are simply mRNA molecules wrapped in a variety of different phospholipids. And we've learned the names of a few. There are many different phospholipids, and there's some cholesterol in there too to pack it up, like a little LDL particle. And that's what the mRNA vaccine is. And they're just using that same technology to pack up different mRNAs to produce these different vaccines. And so just a quick glimpse, you're not going to be tested on that, except for maybe synthetic biology, and but just the scale of their activity. And there's lots of different ones. This is just vaccine. There are lots of different things that biotech companies are doing. Lots of green strategies, like can we replace all the concrete and the concrete industry and the building industry with reusable, green living cells extract to make existing products with smaller carbon footprints and stuff like that. Biofuels, blah, 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 on and on and on. So that's something to mention too, just roaring ahead. And a very different story in biotech companies today than 30 years ago. Some of them are just rolling forward with lots and lots of cash and doing a lot of work to develop new products across the board. So got to mention that. So systems biology, modern biology, biotech companies, lots of new work. And now we'll just kind of dive into some of these stories about genes and genomes like that. And so the first thing is to talk about this idea of DNA sequencing. DNA sequencing is when you read the nucleotide letters A, G, C, T, like that. And this is just a figure from the book showing a strategy to do that. So this is just the DNA, the genomic DNA. There's lots of it. So how are we going to read it? How are we going to analyze it? And the method is called DNA sequencing, and the instrument is called a DNA sequencer. Like that. And there's a lot of DNA in us. The human genome is 3 billion nucleotide letters long. So we should have a feeling for that. 3 billion nucleotides for a human genome. And we'll use that as a reference point. So we can go by decade and I'll just make these numbers up. But like in the 1950s and prior to the 1950s, there was 0 DNA sequence read by the entire planet. And then in the 1960s, I'll say there was still 0 DNA sequence read, but scientists had read in the 1960s worldwide a few hundred nucleotides of RNA. And maybe at the end of the 1960s, maybe a little DNA, but just like a few nucleotide letters. Again, 3 billion for just one human. And then in the 1970s, 2 scientists won the Nobel Prize for developing DNA sequencing strategies. And one invention that prevailed is still very, very common today. And the strategy is we're going to read the DNA sequence in a test tube by copying it and being able to read the letters that get added. So that's a strategy. Just to mention it once, the other scientist who won the Nobel Prize, his strategy was going to read the letters by chemically breaking the DNA and looking at the pieces. So you can think about that, right? How are you going to read these nucleotide letters one at a time? And one strategy that fell by the wayside was use the chemistry to break it up and see the different pieces. And the strategy to succeed is make a new strand and be able to detect the letters. And initially that was, that even though that was in the 1970s, it was very slow to read the DNA sequence that way. And so in the world, in the 1970s decade, maybe a few thousand nucleotides were read total, tens of thousands total. And then the 1980s, a couple of companies built and marketed DNA sequencers. So an instrument that would get the job done. You just had to put the reagents in and it gets the job done. So 1980s, big jump up. So like 1980s, millions of nucleotides read worldwide. All the fruit flies, all the different species, millions, E. coli, bacteria, all the different species, millions and millions of nucleotides read in the 1980s. Then it increases the 1990s. So it's in the 1990s where the government says, let's sequence the human genome. And lots of scientists are arguing about that because they're like, whew, that's going to be really expensive. They estimate to sequence one human genome, it costs $1 billion. Just one. That's how hard it is. And their strategy is just to buy a lot of those instruments and put them all in labs and just rows of those going all the time. Yeah, we'll get it done. And early in the 2000s, they got one genome done for about that price. And then I have a slide, because people like to show this. So here's the cost of a genome, like maybe they say 100 million. The overall project costs more than that, 2,000. And this is just the price to sequence one human genome, and it's going down like that. And it goes down a lot in like 2006, because companies introduce a new instrument that is going to really scale up. And it's kind of a simple adaptation. Scientists had been reading the DNA sequence in individual test tubes, like here's some individual test tubes. And these companies just built instruments where instead of having the reactions occur in individual test tubes, they had them occur in a solution where there were beads in a solution in a tray, and each bead was reading the sequence of one piece of DNA. So you could get like 1 million test tubes worth of DNA read from one run of these new instruments, like that high throughput sequencing they're called. So a big drop there, continue to drop down, like that, okay? And now it's down like several $100s to sequence a human genome, okay? So also in this sequencing, the instruments are available, so the scale of that activity goes way up, like that. So, when you hear about COVID variants, it's because they're just sequencing, gathering it, and they're just rapidly sequencing, because the genome was just tiny, and they could read 1 million COVID variants a day, like that, right? So lots and lots of sequencing. I just Googled it, and AI estimates that to date now, in the history of humanity, about over a quintillion nucleotide letters have been read. And that's like a billion. So you can't imagine a trillion, but a quintillion is 100 or 1 million billion, like that, so none of us can even think about how much DNA sequence has been read, and it's all being held on supercomputers. One way to think about DNA sequencing is it really parallels the development of computers, right? Like when I was in high school, I interacted with a computer once, and it was the stupidest thing, because there was no screen. It was just all being typed on, you type in something, it would type that, and it would send it away, and then the computer would type back to you, like that. Just junk like that, right? And and it's just gone up like that. And that really has been paralleled like that. And computers really like analyzing data like DNA sequence. It's linear, and they can just go all day on that, just massive. And so we want to talk a little bit about that, because none of us can even conceptualize how much data there is and how it has affected biology. So this is examples of system biology, and so not only do we collect big data, but it really shifts the thinking of big data. So you don't need to learn these terms. Well, you can learn these terms. So there's traditionally people called the sequencing that was being done and is still the dominant sequence, they called it short read sequences. Because like whether it's in tubes or beads on a plate, you're just reading a short piece. And I'll just show you what that looks like here. It's just showing, take a genome, break it up into little pieces, and read each of those little pieces, and read lots and lots of them, and just find the overlaps, and you've got it done. And real recently, completely different strategies have occurred or developed. And so there's a company called Nanopore that does this long read sequencing. And that company takes the DNA and drags it through a pore, and as it's being dragged through the pore, it's reading the nucleotide letters. So really different strategy. And that's great. And so just recently there have been some transformations in the strategy. And those are just increasing the rate, increasing the quality, like that, okay? And and we're doing great, and we have a couple more minutes. So I'll just show you this, just to figure you don't need to know it, but let's just remember, human genome, about 3 billion nucleotides long. You'd say, where's the pressure to read human DNA genomes faster? And the answer is premature babies. So there are some hospitals with really fancy neonatal care units, and there'll be a premature baby born, and they want to get all the information about this newborn to figure out how to help the baby. And one thing the doctor can ask for now, right, because they're asking for many expensive tests, and they're just, let's get this baby's genome sequenced today, So search through this baby's genome to see what kind of alleles they have, what kind of concerns we have about caring for this baby. It's just another assay now, and they have that now, where at some of these neonatal units, the baby's born, or even before the baby's born, they'll just get a little cells from it while it's still in the womb, and they're like, we're concerned about this, and they'll just say, we'll sequence the DNA and see what the issues are for this baby, like that. So very interesting, but still, 3 billion, that's a lot, right? And so 3 billion, and this is not the biggest genome, this is just showing that there are plenty of animals and plants that have bigger genomes. So there isn't a good correlation between, we'll say, complexity and genome size. Some genomes are bigger. And so we want to make a comment on big genomes. And and so we want to talk about that. Given time on Thursday, we want to talk about, like, why is the genome as big as it is? It's really big, and why is it big, and why would some animals have bigger ones? Then there's some small genomes. So like here's E. coli, it's about 1 million. So it's bacteria, much, much smaller. Not surprising, but it's a lot smaller. 3 billion versus 1 million is a lot smaller. And then there's fruit flies, and fruit flies are like hundreds of millions, like that. And yeast. And so we'll look into that and look at, from a systems level, a little bit about genomes, and then start diving into the genomes, because there are a couple of comparisons to make about that. And in doing so, we'll keep trucking on these idea of that systems biology and how it brings a next level function. Oh, we got to hand these back, okay? So let me turn this off and get these handed back. So again, you just have a